DNA is replicated during S phase of interphase. This video zooms into the process of how DNA is copied. This is video 10.6 on DNA replication. In this video will cover these 6 unit goals. If you want a closer look, pause the video now. Even though this video is about DNA replication, it's worth taking a look at the history of DNA in the first place. Watson and Crick famously described the structure of DNA in 1953, but their work was largely built on putting together work from other people's experiments. One set of data that was important for Watson and Crick was the data of Erwin Chargaff. Chargaff looked at DNA of many different species and even different organs from the same species. In this data table, we have some bacteria, E. coli, pneumonia bacteria, and tuberculosis bacteria. There's yeast, sea urchin, herring, a fish, a rat, and he even looked at the organs, different organs from humans, thymus, liver, and sperm. And when stepping back and looking at the data, Chargaff saw some patterns, but couldn't really explain them very well. First, Chargaff found that DNA composition differs among species. All species have DNA with A, C, T, and G, but the amount of each changed between different species. For example, E. coli don't have the same percentages as pneumonia bacteria or tuberculosis bacteria. The pattern that really helped explain DNA structure, though, was that in all species, the percentage of A always equaled the percentage of T, and the percentage of C always equaled the percentage of G. This pattern helped Watson and Crick solve a critical component of the DNA structure. Chargaff's data helped Watson and Crick figure out that DNA is a double-stranded molecule where A matches up with T and C matches up with G. Another huge piece of data came from the work of Rosalind Franklin and Maurice Wilkins. The photo in this picture is arguably the most famous photo in biology. It's called Photo 51, and it's the photo that helped Watson and Crick figure out that the shape of DNA is a double helix. The photo was made by Rosalind Franklin using a technique called X-ray crystallography. X-ray crystallography is a really tough technique, and Rosalind Franklin was among the best in the world at it. What you need to do is prepare a sample in a really specific way. You see here a DNA strand mounted to a piece of cork. Now, preparing the sample is tough all on its own, but then you need to blast some X-rays through this sample at just the right angle. The atoms in the sample molecules scatter the X-rays in different directions, and then the X-rays land on a special type of film. Based on the pattern of where the X-rays land, scientists can work backwards and figure out the shape of the structure of the molecule. Rosalind Franklin created this beautiful Photo 51 of DNA, but just didn't pull out all of the pieces of information and put those together. But Watson and Crick did put all of the pieces together using Chargaff's data and Photo 51 and analyzing another early model of DNA that proved to be wrong. Watson and Crick came up with their famous model of DNA with the structure that we accept today. Immediately when Watson and Crick came up with their model, they saw how the structure of DNA matched up with the function of DNA. One of the functions of DNA is to be copied to pass genetic material from one cell to another. The copying of DNA is called DNA replication. Because DNA bases match up, copying DNA is as simple as unzipping the DNA and making a copy of the code that you see by matching up the opposite base. We now know that the process of DNA replication is a process that's called semiconservative, which means that at the end, each new double-stranded DNA has one original strand and one new strand of DNA. But scientists weren't sure at first how DNA replication worked. Originally, there were 3 competing hypotheses for how DNA replication worked. In this diagram, the dark blue DNA strands represent the original DNA, and the light blue DNA strands represent newly made DNA. One hypothesis for DNA replication is conservative replication. In conservative DNA replication, the original double-stranded DNA would unzip for copying, but then the original DNA would rematch up with its original partner and the newly made strands would match up with each other. Another hypothesis is semiconservative DNA replication. In that one, the original double-stranded DNA would be unzipped. Each original strand is used as a template for a new strand, but then the new strand stays coupled with its partner original strand. Finally, there's dispersive DNA replication. In this model, DNA replication is kind of a patchwork process where each newly made double-stranded DNA ends up being a patchwork of the original DNA and the newly made DNA. One critical experiment set the record straight of which one of these is right. In 1958, Meselson and Stahl used radioactive nitrogen to figure out how DNA replication worked. What they did was grow bacteria at first in an environment with only nitrogen-15, which is heavy nitrogen, as the nitrogen source. What this means is that when bacteria incorporated nitrogen into their nitrogenous bases, there was only heavy nitrogen. After all the bacteria had DNA with only heavy nitrogen, then Meselson and Stahl switched the bacteria to an environment with only nitrogen-14, light nitrogen. What this means is that any newly made DNA would then have light nitrogen in the nitrogenous bases. And since bacteria take about 20 minutes to divide, they analyzed bacteria 20 minutes after switching the nitrogen, and this would be the first replication, and then they let another batch go for 40 minutes to analyze what happened during the second round of DNA replication. They could see the composition of DNA by isolating the DNA from the bacteria, doing a few techniques to separate the DNA based on the type of nitrogen, and then exposing the isolated DNA to a film that detects radioactivity. And what you can see by these data samples is distinct bands of DNA. So you can distinctly see DNA that has two light strands on top, nitrogen-14, nitrogen-14, and this means that both strands in this double-stranded DNA are newly made. The bottom band, which we can see just over here, is for double-stranded DNA with only heavy nitrogen, meaning that both strands are original strands. Finally, here and here, you can see bands that are hybrid DNA, double-stranded DNA, meaning that one of the bands is the original and one of the bands is newly synthesized. Let's match up their data with these models. The original DNA up here was heavy-stranded only. That made sense. Before the experiment started, bacteria only had a heavy nitrogen, so this was kind of like a control. After one replication at the 20-minute time point, they saw one band, and that band was only hybrid DNA. This means that the double-stranded DNA, after one replication, was a mixture of original and newly made DNA. This piece of data right here ruled out conservative DNA replication. That one is not the right one. And narrowed down the choice to either semiconservative or dispersive, where the DNA is hybrid. After two replications, they saw two bands. One where it was newly synthesized DNA only, light nitrogen and light nitrogen, and another of hybrid DNA. This means that some of the DNA double strands had to be made of only newly made DNA, and the other had to have some original DNA and some newly made DNA. The light nitrogen only band, this one up here, ruled out the possibility of dispersive, since all of these are hybrid strands. And this is how the experiment steered scientists towards understanding that DNA replication is semi-conservative. Studying the molecular biology of DNA replication is a story of finely tuned orchestration between 5 different enzymes: Helicase, Primase, DNA Polymerase III, DNA Polymerase I, and Ligase. DNA helicase is the enzyme that unwinds and unzips DNA. Unzipping the DNA exposes the nitrogen bases to get copied. DNA is held in its open conformation by these proteins right here called single-stranded binding proteins. The spot where helicase opens up the DNA and the DNA replication is taking place is called the replication fork, because the DNA fork's right there. DNA helicase knows where to go because there are starting points on the chromosomes called origins of replication. To make the replication process go faster, eukaryotes have a whole bunch of origins of replication all along the chromosome. Also, DNA replication happens in both directions from the origins of replication. So a better name for the replication sites are replication bubbles, but you can see that each replication bubble has like 2 replication forks, one on each side of the replication bubble. Well, this diagram shows DNA replication in action, where as replication continues, the replication bubbles get bigger, and eventually those bubbles will merge until you have complete copied DNA. This diagram over here is a photo of actual DNA replication in action, and you can see the arrows pointing to 3 replication bubbles in this picture. Helicase opens up the DNA to create space for the builder enzyme, DNA polymerase III. This slide describes the building process as complementary bases are added to the 3' end by DNA polymerase III. Although this sentence is brief, the sentence isn't so simple, so let's dissect some of the words that are in there. First is complementary. It's really important to understand that in DNA, complementary refers to how A matches up with T and how G matches up with C. DNA polymerase III uses these chemical rules when it makes a new DNA strand using an old strand as a template. Next, what's this 3' all about? Well, 3' is a chemistry term that refers to the carbon numbers in deoxyribose. So in deoxyribose, the carbons are numbered 1', 2', 3', 4', and 5'. On a nucleic acid, one end of the nucleic acid has the 5' carbon pointing out, and the other end of the nucleic acid has the 3' end of the carbon pointing out. DNA polymerase III can only add new bases to the 3' end of a new strand of DNA. The way double-stranded DNA molecules are set up is in an anti-parallel way. What this means is that one DNA strand goes from the 5' direction to the 3' direction, and the other DNA strand goes in the opposite way, where the 5' is down here and the 3' is up there. This creates a weird scenario for DNA polymerase that can only add new nucleotides on the 3' end of a growing strand. One way that I can tell the 3' and the 5' ends of a DNA is I think of the sound of 5' and phosphate. So the end that has the phosphate is the 5' end, and that has the phosphate is the 5' end. And then dissecting this statement, I want to take a closer look at DNA polymerase III. Since DNA polymerase III is an enzyme, let's think about it as an enzyme. The substrates that fit in the active site of DNA polymerase III are the growing nucleic acid strand and the nucleotide that's about to be added on. The reaction that's sped up by DNA polymerase III is building DNA polymers. In other words, it's an endergonic anabolic reaction, and DNA polymerase III joins together nucleotides by forming phosphodiester bonds on the 3' end of a growing DNA strand. So where does the energy come from for these endergonic reactions? Well, it's pretty cool. When the nucleotide monomers come in, they come into DNA polymerase III in their triphosphate form. DNA polymerase can couple the energy from exergonic breaking of these phosphate bonds and use that energy to power the endergonic reaction of building DNA, where only one of the phosphates ends up in the final nucleic acid. DNA polymerase III is a really powerful enzyme, but DNA polymerase III has its limitations. One limitation is that DNA polymerase III can only add nucleotides to a growing DNA strand. Well, what that means is that DNA polymerase III can't start a DNA strand. So how does a DNA strand start? Well, for that, we need another enzyme. The enzyme that creates a starting place for DNA polymerase III is called primase. Primase goes to the spot opened by helicase and creates a small primer that's made of RNA nucleotides. A primer is just a short starting sequence. It's okay that the primer is made out of RNA. There's another enzyme that will take care of that later. Another limitation of DNA polymerase III is that DNA polymerase III can only add bases to one end of the DNA strand. What this means is that in a replication bubble, the strands are made differently. One strand is made continuously in one long piece. The other strand is made discontinuously in several smaller pieces. The strand that's made all at once is called the leading strand, and the strand that's built a bit at a time is called the lagging strand. Let's look at the leading strand. Now, when this replication fork was first opened by helicase, it was opened, say, right here. Primase came in and made this green primer, and then DNA polymerase III was able to find its starting point and build this red strand of DNA. Before DNA polymerase gets to the end, helicase is going to open up more of this helix for DNA polymerase III to copy. This new red strand will go all the way to the end as one long piece. On the other strand, though, DNA polymerase III is going the opposite way. When the double helix was opened up, primase made this green primer right here. DNA polymerase III found its starting point and started building a DNA strand the other way. Then, as more of the helix was opened up down here, primase came in and made this next green primer, creating a starting point for another DNA polymerase III. This DNA polymerase III cruised along and made this red strand of DNA until it hit the other primer. This process of making DNA in small bits is called discontinuous DNA synthesis. As more of the helix is opened up, primase will make another primer, creating another starting point for DNA polymerase III, and DNA polymerase III will fill in the hole, and so on and so on. These small DNA pieces are called Okazaki fragments, named after the Japanese scientist who first described this process. Now it's on to our fourth enzyme, DNA polymerase I. DNA polymerase III is the workhorse builder of the group, and DNA polymerase I kind of cleans up everything after DNA polymerase goes through. First, DNA polymerase I takes out RNA primers and replaces those RNA primers with DNA nucleotides instead. You can see DNA polymerase III doing the job of building. DNA polymerase I fills in and replaces these RNA primers. Next, a DNA polymerase I can also proofread and correct mistakes that DNA polymerase III made. Here's the proofreading mechanism of DNA polymerase I in action. DNA polymerase III makes about one mistake for every 10,000 nucleotides. That doesn't sound like a lot, but in humans this would mean that every time the DNA is copied, DNA polymerase III would make about 300,000 mistakes. For each mistake, DNA polymerase I finds the mistake, cuts out the bad nucleotide, and replaces the bad nucleotide with the right one. Having DNA polymerase I reduces the error rate to about 1 in a billion, but this means that there are still mistakes every time your DNA is copied. These mistakes are called spontaneous mutations and are one of the causes of cancer that we talked about in the last video. The final enzyme in the story is DNA ligase. Once the polymerases are done, DNA ligase goes through and joins together all of the DNA fragments. So let's recap. This video gave a little bit of history of science and explained how Watson and Crick first described the DNA structure after putting together a lot of different data from a lot of different scientists. Rosalind Franklin, Maurice Wilkins, Erwin Chargaff, and Linus Pauling. Second, a DNA replication is semi-conservative, and we looked at the Meselson and Stahl experiment that kind of worked all of that DNA replication mechanism out. Finally, we talked about the molecular biology of DNA replication, a finely tuned coordination between 5 enzymes: helicase, primase, DNA polymerase III, DNA polymerase I, and ligase.